Now it's my pleasure to introduce Angela Belcher, who is a biological and materials engineer with expertise in the fields of biomaterials, biomolecular materials, organic, inorganic interfaces, and solid-state chemistry and devices. Her primary research focus is evolving new materials for energy, electronics, the environment, and medicine. Angela currently holds the Keck Chair in Energy at MIT and is faculty in the Department of Biological Engineering, Materials Science and Engineering, and the Koch Institute of Integrative Cancer Research. She has a really stunningly long list of impressive awards that she's received. She's a MacArthur Foundation Fellowship winner, the recipient of the 2013 Lemelson MIT Prize, an elected member of the American Academy of Arts and Sciences, and was listed by Rolling Stone magazine as one of the top 100 people changing the country. She's founded companies including Cambrios Technologies and Siluria Technologies, and she's been a dedicated educator at all levels, having mentored 39 PhD students, hundreds of undergraduate students, as well as working with thousands of kindergarten through high school students and teachers. So it's a real pleasure to welcome Angela.
Thank you very much. Well, thank you for the invitation to be here, and it's great to follow that wonderful lecture from before. I also work on phage and carbon nanotubes, so it's a nice transition. So I've spent my career evolving organisms to grow new materials for electronics and energy mostly. My dream has been to give genetic information to organisms so that they can grow and assemble non-biological structures and improve device performance. And so I'll tell you a little bit today about how to think about going from making solar cells and making batteries and working in the oil and gas industry to thinking about designing new materials and devices for medicine. Specifically, over the last 2 years, we've been working on developing new technologies to find tiny ovarian tumors for guided surgery to try to help the surgeon find smaller tumors earlier on to increase the survival of patients. Right now, we're working in mice. And so this is part of the team that I'm going to present data about today in terms of trying to develop new probes and new imaging technology for looking what's called the second window near-IR imaging. So imaging with light above 1,000 nanometers to try to look deep inside, deep meaning about 10 centimeters inside of an organism for very small tumors. And it's a collaboration between groups at MIT, specifically on the ovarian tumors, Professor Sangeeta Bhatia at the Koch Institute, MIT Lincoln Labs, and Mass General working with Michael Beer on oncology. And so like I said before, the focus of my research over the last several years has been trying to understand how biology makes materials. Examples are bones and shells. So about 500 million years ago, organisms in the ocean started making hard materials. They adapted the proteins they had for other functions and started grabbing calcium and carbonate or phosphate out of the environment and started building beautiful nanostructured materials, which became things like abalone shells, diatoms made out of silica. And so we focused on saying, wow, organisms did such a fantastic job with a limited number of materials. What if they had the whole periodic table to work with? Could you find a DNA sequence that coded for a semiconductor? Could you find a DNA sequence that coded for a battery electrode? And then you could actually use that to assemble the materials, like you heard of the manufacturing, but instead of manufacturing a material, manufacturing a battery. And we've worked on that over the last couple of years, or manufacturing a solar cell. Well, organisms in the ocean haven't had the opportunity to work with all those elements on the periodic table, so we use a combination of selection and rational design to find peptide sequences that recognize non-biological structures, can grab onto them, maybe grow them, assemble them, and deliver them where we want them to be in a device, or in this case, targeted to ovarian cancer. And so I'm also really interested in nanoscale, and I'm not interested in nanoscale necessarily because it's small, because really beautiful things happen at the nanoscale. When you take just a small number of atoms and you start to control where those atoms are, what their shape is, really interesting things happen. And you get changes in strength of materials, you get changes in electronic properties, magnetic properties, optical properties of materials at the nanoscale. So that's why we're interested in it. And we think that biology is really the really great tool to control things at the nanoscale, because we are nanoscale objects, but scaled to the meter scale. And proteins and DNA, antibodies in our body are already at the nanoscale. scale. So using those as a template to grow materials is what we're interested in. And so I've always said I've never been accused of being focused because we basically take nanomaterials, control their structure, and work in all these different fields, from solar to batteries to fuel, from CO2 sequestration storage, water purification, and then just recently imaging and therapeutics. And so about 3 years ago, I was asked to join the Cancer Institute at MIT. Having never worked on cancer before, they said, but can you take some of the same tool sets you've developed in electronics, and can we see how they could be integrated in for cancer research? And so again, we're fascinated by biomolecules because they're nice and small. This is how biology, how nature's evolved to grow materials, inorganic materials, is by using biology as a template to grow those materials. And this is an example of an abalone shell, which is calcium carbonate. It's a biocomposite material. You might have pearls. Those are 98% by mass calcium carbonate, 2% by mass protein, yet they're 3,000 times tougher than their geologic counterpart. It's all encoded in their DNA sequence. When a male and female abalone reproduce, they pass on the genetic information that says, this is how to build an exquisite material. Here's the exact map on how to do it. So that's our inspiration. And so what we set about to do several years ago now when I first started was to try to find a protein sequence that codes for the assembly of any material we want. And today we're going to talk mostly about carbon nanotubes. And so how do you get, it took about 50 million years for organisms in the ocean to get good at making and printing inorganic materials. We have, on the time scale of a PhD student, 4 or 5 years. And so we're using phage display as well. We're choosing proteins to be able to bind a material. So to material scientists, a phage is a wonderful material. It's 880 nanometers in length and 6 nanometers in diameter. And the proteins that make it up are encoded by its DNA. So you can make libraries of these. You can change the minor coat protein on the end. You can change the major coat protein that surrounds it. And so we look at it as this beautiful scaffold that's genetically engineerable to be able to bind anything that we want to be able to bind through a process of selection and evolution. And this is just showing what the structure looks like. The modified proteins on the surface could be things that could be done through random mutations or rational design. And you make libraries of proteins. And you're going to take those libraries of proteins and get them to bind to and assemble or grow a material of interest. And so the basic idea is you start with a billion possibilities. You force them to interact with anything that you want to. And you heard in the last really great talk how they were used to pull a specific biomolecules or drugs and concentrate them. What we want to do is actually get them to bind semiconductors, insulators, metals, and carbon nanotubes, and be able to assemble those kinds of materials. And so there's some similarities. And the other thing that's really hard to do is actually mix things on the nanoscale. Because you can either think of mechanical binding or chemical modification, but if you have a really precious material, you may not want to change its chemistry too much on the surface because you may change its optical and electronic properties. And so we think of these M13 phage as mediators that you can genetically control to bind to one material, assemble another material, and then deliver to a separate location. And so we think of this bacteriophage as the ability to be able to bind to carbon nanotubes. Carbon nanotubes in solution basically are usually surrounded by a surfactant. And we said, well, can a phage be a surfactant? Can you select or evolve a bacteriophage to grab onto one of these little nanostructured materials and protect it and be able to deliver it where you want it to go? And we showed that you could do this for batteries. This was a battery that went to the White House. It was a record-breaking battery at the time. It was genetically engineered, genetically evolved battery. We showed you could do the same thing for solar cells. But then we said, okay, we're really good at working with these carbon nanotubes. Now, carbon nanotubes are graphene sheets of carbon that are basically rolled. And they can be rolled at different diameters, give them different chiralities, and those give them different optical electronic properties. Well, we started thinking about the fact that we showed these are great in electronics, but they can be interesting in optical materials as well because they're fluorescent in an area that's called second window near IR, which is about 950 nanometers to above 1,400 nanometers. And it's an optically transparent region. It's transparent to blood and tissue. Now, there aren't very many optical probes in this area that would allow you to image deeply into the body. But carbon nanotubes are one of those possibilities. Now, in this second window near IR, there's much less tissue autofluorescence, and so you could also, there's less scattering, and you can improve your signal to noise by about 100-fold. And so what we said was, well, maybe you can develop something that's, that you can see deeply into the body at long wavelengths, up to about 10 centimeters, that's non-radioactive, inexpensive, and quick imaging. And you can basically do this with light compared to other more costly procedures. And so that's what I'm going to focus on in the rest of the talk. And so there's trade-offs between resolution and depth, from high resolution at shallow depths to much lower resolution as you go deeper, but we wanted to be able to do both. And the particular cancer that we focused on in the beginning was ovarian cancer because it's difficult to diagnose. It's often diagnosed late, and it doesn't have a great survival rate. So we were working with Dr. Beer at MGH on developing probes for small ovarian tumors. And so the gold standard for this right now is contrast-enhanced CT, and it has poor sensitivity for small tumors. And from the surgeon's point of view, optimal debulking of these tumors gives the best survival in patients. And so the primary care for this is debulking followed by chemotherapy. And our idea was if we could find smaller tumors and if we could find hidden tumors, that it would increase the survival of the patient. And so what we set to do was build a set of imaging tools in our labs in collaboration, as well as new imaging probes. And I'll show you some of those right now. So to start with, we used M13 bacteriophage. We genetically engineered it to pick up single-wall carbon nanotubes, which are going to be fluorescent in the second window near IR. We engineered it on its end to have targeting ligands that bound to the molecule SPARC, which is overexpressed in ovarian tumors. And in fact, SPARC has a low expression in healthy ovarian tumors and has a high expression in carcinomas. And so this high SPARC expression is correlated with poor prognosis. And we looked at carbon nanotubes as well because of their interesting optical properties, but we also showed that they didn't quench and they stayed stable under the time period for surgery. And so we built our first generation imager, which is an indium gallium arsenide detector and an 808 laser, and we started in mouse models where we could do screening, we could do mouse, and we can do phantoms. And what we found is that looking in the second window near IR, that you could find tumors, and this is in, my laser pointer's not really working here. Oh, here it is, okay. That we could find tumors in the second window near IR that you couldn't find in the first window near IR or in the visible. And so we had actually much better tumor to muscle and tumor to background ratio with these probes in our mouse model in this ovarian cancer model. And we could find tumors that we couldn't see by other imaging methods. This is a mouse that has a heavy burden of ovarian tumors, and this is before injection, and there's no background. After injecting the probe to the intraperitoneal space, it starts to pool. In about 24 hours, you get optimal imaging. We have about 10-fold increase from tumor homing. These are phages that are binding carbon nanotubes that are directed towards these tumors. And we have a much better target to background ratio. We can look at these nodules afterwards, and we can see the fluorescence in the tumors that were removed. We don't see any binding to non-specific organs, and we can see them in actually organs that the surgeon couldn't see beforehand. So this is one that was hard to see, and you can see it in our fluorescent imaging. And this is a second one that was hidden by other organs, and you can see it in the second window near IR imaging system. So we did a series of experiments where we gave the surgeon a map. This is pre-injection. This is what the map looks like, where we think all the tumors are pre-surgery. He goes in without our map, and he removes all the tumors that he can see. And then he has our map and goes in and removes the tumors that he couldn't see by eye. And what you'll notice here is that he's a very good surgeon. He can see the tumors in this area, but he really couldn't see the tumors that were 1 millimeter or below. And so having the surgical guided system allowed him to find much smaller tumors. This is just showing that he could find 30 versus 4 submillimeter tumors using this map. And actually, he could find tumors as small as half a millimeter. Then we decided to actually make a real-time guided system to where the surgeon could get real-time information about size and distance of the tumors. And a lot of this progress also had to do with development of algorithms and the software that was developed by MIT's Lincoln Labs to guide the surgery. And so this is the raw image. This is a modified algorithm, and this is nonlinear remapping, where a lot more information can be pulled out by the surgeon. And we can go and say, okay, can you separate these before the surgery starts into size of tumors? And so here we have ones that are 1.5 to 2.5 millimeters in between, and they're very small ones. So he or she can make a decision before they go in about the ones that are operable and the ones that are too small. And then using additional algorithms developed, we can also estimate the depth of the probe before it starts, before the surgery starts. We have a tumor specificity way of being able to tell how much of the tissue to take out. This is tissue that's removed from the animal during surgery. This is 2 millimeters. We can break that down and say, of that 2 millimeters that were pulled out, how much of it was really tumor and how much of it was healthy tissue. And using this, we've been able to say that about 50 of the excised tissues, 49 of them were confirmed to have tumors. So we have a pretty good diagnostic tool as a pre-surgical and during surgery in these mouse models. So we're looking at much deeper. Those were only in mice, which are on the order of a centimeter or so. We've been able to look at up to 9 centimeters using other kinds of probes, and then developing what's now our 4th generation instrument. We hope to do tracking in real time of individual tumor cells, as well as develop inexpensive hardware and software to get basically 3-dimensional information, inexpensive, minimally invasive, and we want to be able to track a single cancer cell or an immune cell or an infection agent in a whole body in real time. So that's my story of how you get from solar cells and batteries to developing new tools for ovarian cancer diagnostics. Thank you.